Hi, my name's David Simpson. I'm a reader in the Wellcome-Wolfson Institute for experimental medicine in Queen's University, Belfast, and I work in vision science. So today I'm going to talk to you about some of the work we've been doing using single-cell RNA-Seq to understand the cellular basis of several eye diseases. So hopefully, after hearing about the various examples I'm going to talk to you about, it will help particularly people who are new to the field guide their use of single-cell RNA-Seq in whatever system it is that you're interested in. And hopefully it'll also pick up something about the basis of various eye diseases. So I'm going to be talking to you today about three different conditions. So starting up at the top there, diabetic retinopathy. So this condition is the most common cause of vision loss amongst those of working age. And it affects both the vasculature in the retina, the back of the eye, particularly the microvasculature, but also the neural retina as well. I'll also talk about glaucoma. So this can cause permanent vision loss due to damage occurring to the retinal ganglion axons as they leave the eye and the optic nerve. And finally, I'll talk about age-related macular degeneration, which is a terrible condition which causes loss of your central high acuity vision. And as the name suggests, this is more prevalent among older people. So approximately 5% of those over 65 and over 10% of those over 80 are affected by age-related macular degeneration. So we'll start with diabetic retinopathy. And as I mentioned, this is often thought of as perhaps primarily a vascular disease. So we get dysfunction of the microvasculature, which becomes leaky and can lead to a buildup of fluid in the back of the eye, or edema, which can cause vision loss. And then in the later stages in advanced diabetic retinopathy, we can actually get proliferation of the vessels, which causes major loss of vision. However, there's also damage to the various cell types in the neural retina. And this is perhaps less well characterized. And there's something of a debate as to how the two are linked. Do the problems with the vasculature cause the neural damage, or does it happen independently, and which happens first? So this is the question that we tried to address with our study. So we used a mouse model of type 1 diabetes, in which we use streptozotocin injections, which destroy the beta cells in the pancreas, and the mice become diabetic. So after 3 time periods of 3 weeks, 4 months, or 6 months, so I'll refer to that as early, mid, and late periods. So this is length of diabetes. We sacrificed the mice, dissected out the retina, and then isolated the individual retinal cells, which you can see in the image in the bottom right. So this took some optimization, because this is, as I'm sure you'll hear from other speakers, this is really a critical step in single-cell RNA sequencing experiments. We need to have high viability cells. But even with this, there will be some damage and RNA spilt from the cells, so there'll be ambient RNA, which is particularly a problem in retinal studies. So these experiments were performed using the 10x Genomics platform. So we analyzed the data using a standard cell ranger from 10x and Surat version 3 pipeline with the normal quality control steps. And you can see from this UMAP image of the different cells. So I should say we've approximately 10,000 cells here, and each dot in the UMAP is an individual cell that are clustered together according to similarity of their transcriptomes. And we've got quite nice separation of all the various cell types. So you can see in the cartoon diagram on the right-hand side what those different cell types are. And this is adapted from the seminal paper by Micosco et al., which was the first group to use a drop-seq to analyze the retina. So we were quite happy with our initial results. So we've got very nice resolution of the different cell types. This kind of raises the question of how many actual cell types or clusters of cells are there? What is the best way to define the different cell types? So we used initially the default settings on Surat, and we have approximately 50 different clusters, which you can see indicated here. But this number can change depending on the settings, and there are some ways you can try and determine the optimum number, but in some senses it's up to you to decide the granularity of your clustering, and this you need to think about depends on your question. So all these multiple cell types are very useful, and we've looked at those, and it provides some information about the potential roles of the different subtypes. So in the default settings, as I say, we had about 50 different clusters, and by looking at the marker genes, we could assign those in the middle UMAP there to the different cell types, so the subtypes of the main classes of cells, amacrines, bipolar, and so on. So as I say, this is very useful, but one thing that we became aware of is that as you make smaller and smaller clusters, you have less cells, and this then gives you less power to look at differential expression. So sometimes it's beneficial to do a more coarse clustering, as we have on the right-hand side, where we group, for example, all the rod cells together, and then we can perform differential expression between the control and the diabetic, and have more power to find differentially expressed genes. But before even considering actual differential expression of genes, one of the great powers of single cell analysis of a complex tissue is that we have the numbers of cells from all the different cell types, and we can look at the proportions of those individual cells, and that's what's shown in this visualization here. So just to orientate you, so each of the dots here represents an individual cell, and then if we look vertically, we have each of the cell types, the amacrine, bipolar, and so on. And then the two rows indicate the control cells and those from the diabetic mice. So as you can see, the patterns are very similar. So we've obviously the same cell types, but the number of each subtype of cell within those, indicated by the different colors, are also fairly similar. So in fact, we had no significantly different changes in proportions. And if we just focus now on two of the cell types, so on the right-hand side, you see the rod cells, and there are slightly fewer in the diabetic retina, although it wasn't significant with the numbers that we had. But if we now look at the size of each of the individual spots, so remember each spot or dot represents a cell, but the size of that is, in this case, is being determined by the number of features, so the number of different genes being expressed by those cells. This is a reflection both of the cell size, so if they're small, obviously you'll have less genes, and also the diversity of genes being expressed. So in the rod cells that we see highlighted on the right, they're very small dots, because these cells are, and the cell body anyway is very small, and also they express a small range of genes, but to a very high expression level, whereas the ganglion cells that are outlined there, each individual spot is bigger because they're larger cells with a wider range of genes expressed. This is just reminding you there's extra information available from single-cell RNA-Seq data that you can look into. Coming back to the differential expression, so now this is at each time point we had three 3 mice from the control and diabetic. So we separated out the cell types and then looked at the differential gene expression. And I'm just showing the results from some of the more abundant cell types because it was difficult to compare with the rarer cell types. Unfortunately, there were too few vascular cells to give very significant changes. But if we look, we see the different cells seems to be responding in different ways. So the rods, for example, have most changes at the later time point, indicated in red in the diagram, whereas amacrine cells seem to be affected their gene expression early on at the early time point, and then not so much later on. And again, the Muller cells have most changes later on. So if we just focus on that a little bit more, so the amacrine cells at the early time point. So I've pulled out the most differentially expressed genes with most significant changes, and these were almost entirely fell into these 2 groups, which indicated with the green surround are components of the ribosome, and each of the genes in this network, the color indicates the relative expression between in the normal and the diabetic. So the blue color means that the various ribosomal genes have gone down early on in amacrine cells, so suggesting that they're reducing their protein synthesis, whereas genes associated with the mitochondria are increased in expression. So presumably there's more electron transport and ATP generation occurring. And if we compare this with the later time point, with the 2 cell types I mentioned before, the Muller glia and the rods, so again we have changes in gene expression of the mitochondria genes, but at this time point they are reducing in expression, and there's less obvious changes in ribosome. But as you can see in this small network, there are many other genes also affected, so there's more diverse effects at the later time points, as one might expect. So we can see that shown in this image. So if you look on the left-hand side, we've looked at, again, at 5 of the more numerous cell types. So the rods, Muller glia, but also the bipolar, the other photoreceptors, the cones, and amacrine neurons. So you can see that there are some functions that seem to be altered in many of the cell types, such as the respiratory electron transport and ATP synthesis that I showed you before, and also protein synthesis functions. But there are also other pathways and types of function that are also altered, but just in some of the cell types, and these are currently under study. So these are as yet unpublished data. So this is also borne out if we look at the Circos plot on the right-hand side. What this indicates for each of the cell types labeled there is the number of altered genes. So the longer the proportion of the circle, the more genes there are altered in that cell type. And the purple lines join the same genes that are altered in different cell types. So I think what this shows is that certain cell types, such as the rod and bipolar and even the Muller glia, have many genes which are altered and fairly specifically in those particular cell types. So although there are shared effects, different cell types are also differentially affected by diabetes. So we'll shift now to focus a little bit more on the retinal microvasculature, which is perhaps the main type of cell affected in diabetic retinopathy. So they're known to become leaky during the pathology. So we want to look in a bit more detail. And you can see from this one image that there is heterogeneity in the expression of certain, in this case, proteins, but presumably the underlying messenger RNA in different cells. So to do this, we used a cell culture model of primary bovine retinal microvascular endothelial cells. And in this case, was using the Fluidigm platform and a full-length transcriptome, which is measured using the SMART-Seq protocol indicated in the diagram on the right. So this allows us to look in a bit more detail about some aspects of the messenger RNA not possible with the 10x platform, which just looks at the 3' ends. So most of this work has been published in Mulvey's. So when we did initial clustering of the cells, they very clearly fell into 2 clusters, if you look on the left-hand side. And there was a small group of cells that were clearly proliferating. But it's possible to regress out cell cycle effects. And if you look at the UMAP on the right, that's what we've done with these cells. And you can see there's a fairly homogeneous population just forming this one large cluster, which seems to not be affected by batch effects from the different animals or the different batches that we ran the experiment in. So a fairly homogeneous population of endothelial cells. So what we're particularly interested in is the barrier function, so the connections between the endothelial cells, because this is what becomes dysfunctional in diabetes. So we used the gene expression data from the single-cell RNA-Seq to see which of the many known potential components of the barrier were expressed. And this diagram indicates those that are expressed and at what level, at least at the messenger RNA. And this is confirmed for some of the genes using immunohistochemistry. So, I mean, it would be possible to generate this model using bulk RNA-Seq data, but because this is single cell, we can extract some more information. I've just got some examples of the barrier genes from the previous slide here and their expression levels. So again, each dot indicates the expression in an individual cell. And you can see there are some genes that are highly expressed in all the cells, and then some in the middle there, the DSC2, for example, are expressed at moderate levels in some cells and not at all in others. So what this allows us to do is then look for correlation between the expression of different genes. So what the correlation on the right is showing us for the barrier genes just is which genes are expressed in the same pattern together between different cells. So what genes are low in some cells and then high in another cell and so on. So follow the same expression pattern. We can see when we look at the most highly correlated genes on the very top right of the slide, you see that red square. So those are a small number of genes that seem to center on PCAM1 that are expressed at similar levels together. So we need to be careful not to overinterpret this, but it at least is evidence, suggestive evidence that these genes may be functioning together, and that this is something we can, then investigate further. So the final bit of data to show you from these experiments is about alternative splicing, because as I mentioned, for this work we have full-length transcripts. So we can then look at splicing right along the length of the transcript. So I've got examples from 2 genes here, AD1 and SNAP23. And I've chosen just 4 cells, illustrated cells, to show you the alternative splicing. So I've highlighted here the exons which are spliced in some transcripts and not in others. So if we look at AD1 at the very top in the first cell, the transcripts all go from the left-hand exon to the right-hand exon. So there's a percentage spliced in value of 0. So in that cell, all transcripts don't have the additional exon. Whereas if we look down to cell 4 in AD1, for example, all the transcripts have that exon spliced in, whereas in cell 2 there's a bit of a mixture. And the numbers on the right-hand side, the 177 at the top there, is the number of reads we have for each event. So I think there's reasonably strong evidence that this is occurring, and it's quite exciting that in some cells we seem to get almost exclusively one isoform, and in other cells a different isoform. And depending obviously on the gene and the exon, this may change the protein that's been encoded. So again, we need to be a little bit cautious with some of these results, because it is difficult to be too certain because of the level of difficulties of measuring changes in an individual cell. But I think this is a very exciting prospect for more studies of alternative splicing at the single cell level in the future. Okay, so now I'm going to tell you a bit about a study where we looked at some of the causes of age-related macular degeneration. And I'd mentioned that one of the cell types involved in that is the retinal pigment epithelium, which are the cells that form a layer at the bottom of the retina. So AMD is multifactorial, and there's many factors involved. But one of the key pathologies is the development of these so-called drusen deposits, which occur between the RPE and the rest of the, and the basement membrane on which they sit at the bottom of the retina. So you can see in the image on the right-hand side, the two stars indicate these drusen deposits, and the dark layer is the retinal pigment epithelium and the neural retina above that. So the RP should form a flat layer, but in this case, the drusen are formed underneath. So my collaborator Imre Lengyel and others have shown that if you grow primary human RP cells long-term in culture, they develop many of the phenotypes similar to that in vivo, and in fact you can model the formation of these drusen. But in these models and in vivo, there's evidence that there's heterogeneity in the RPE cells. So we wanted to use single-cell analysis to understand the pathology and this model in more detail. So this is an outline of the experiment that we performed. So these RPE cells were grown in long-term culture, either for just a couple of weeks, several months, or up to 5 months. And you can see the images of the cells below there, and the longer they're in culture, they become pigmented. But there is this heterogeneity in that some cells are more pigmented than others. And the transepithelial resistance of the cells also increases with time, which is an indication of their mature phenotype. So this is the initial clustering of approximately 20,000 cells from all combined together from those different time points. And you can see we've got a reasonably homogeneous cluster, and the different libraries from the different cell, different times overlap quite a lot, although I'll come back to that a little bit later. So we're reasonably happy with the experiment. There don't seem to be any batch effects, for example, at this stage. So then this came back to the question of the clustering. How many clusters should we have? So using the default pipeline, we end up with approximately 13 clusters, as shown in the bottom right there. But you can vary the resolution of your clustering algorithm and generate fewer clusters. So we were kind of wondering what was the best answer. So we can look at the 13 clusters, and this can provide information about subspecialization of the different cell types. But are they really all different meaningful cell types? So we then looked at the expression of some known RP genes, and there appear to be two main groups. So the sort of left-hand area of the plot or cluster seemed to be high expression of the RPE genes, known RPE genes. And if we also look then at the libraries in terms of the time points, we see that the long-term culture indicated in blue in this plot seems to be more in the left-hand part of the plot, which seems to again correlate with the more mature phenotype. So to investigate this further, we generated a list of genes that have been associated with the RPE, either from the literature or previous experimental studies. And then we performed clustering. So you can see on the heat map on the right-hand side. And the heat map shows the average gene expression for each of the clusters shown in the UMAP there. And these clearly fell into two groups, and the clusters co-locate with the two halves, if you like, of the UMAP. And I've just indicated some of the best known RP marker genes at the top right-hand corner there, and they're located in the what I've indicated, the red cluster, which is the left-hand one in the diagram, which equates to the more mature cells. So in terms of our RPE phenotype, it seems that we actually have two main subclusters. On the right-hand side are the less differentiated cells in red, and then more differentiated is the large cluster on the left. So this was interesting for us, and obviously we can now perform differential expression between these two populations, and this will, as well as showing us the known RP genes, potentially along with that there's some other genes involved in RP function that we're discovering. And also what we looked at was the proportions of these two populations of less differentiated and more differentiated cells at the different time points. So if you look at the graph in the bottom right-hand corner, so we're going from the shortest time point of a couple of weeks up to the longer time points up to 5 months. And you can see that the proportion of more differentiated cells increases with time. So it seems that the more mature phenotype that we generate with long-term culture is not so much that all the cells become gradually more mature, it's that there's a greater proportion of the more mature subtype. So this now allows us to interpret the experiments using this model more effectively. And it also potentially has in vivo relevance, because I haven't showed it here, but there's some evidence that these subpopulations actually exist in vivo. So having these two cell populations also allowed us to study the transition between the less differentiated to more differentiated, so the maturation process, if you like. So for this, we used the pseudo-time analysis using the Monocle software. So what this does is aligns all the cells along this trajectory from the least differentiated to the most differentiated. And we focused on the principal trajectory, which goes from the start point indicated in blue there in the right-hand figure, and winds its way to the most differentiated cells in red on the left-hand side. So all the cells are lined up along this trajectory, and then we can look at how gene expression changes along that maturation gradient, if you like. And this is just an overview of the results. So what we have here are groups of genes, which are described as modules. So each horizontal panel here is the average expression of a group of genes that all have similar patterns of gene expression as we move from less differentiated cells on the left-hand side to the more differentiated on the right-hand side. So we kind of have a pseudo-time going from left to right. So you can see that there's essentially two groups of genes, those that are higher in the less differentiated, so the red ones on the left, the bottom, and genes which are higher in the more differentiated, so that's on the right-hand side at the top. So we could have found a lot of these genes by just comparing expression, as I mentioned previously, between the less and the more differentiated, but this pseudo-time gives us some more information. And perhaps the most interesting genes are those ones that I've highlighted in the middle here, which seem to have increased expression just around the period of the transition between less to more differentiated. So these are the genes that are most likely to tell us about how this transition works, rather than being just characteristic of the two populations of cells. So these are genes that are now under further investigation. Okay, so the final study that I'll just finish off briefly telling you about involves some preliminary work looking at the causes of glaucoma. So as I mentioned before, glaucoma causes permanent loss of vision and reduction in your visual field, and this is due to progressive degeneration of the retinal ganglion cells. So the pathogenesis is not well understood, and it's often but not always related to elevated ocular pressure. A particular area that's come under focus is the lamina cribrosa, which is shown in the diagram on the right. So the retinal ganglion cell axons spread across the surface of the retina and bringing the signals from the visual process out through the optic nerve and back to the brain. So it's where these retinal ganglion cell axons go through the optic nerve at this region called the lamina cribrosa, where it's thought the damage occurs. This is kind of a sieve-like structure which helps to control the pressure differential between inside and outside the eye. So there's some evidence to suggest that part of the pathology is an increase in extracellular matrix deposition by the lamina cribrosa cells, which are indicated just in the top right of the cartoon diagram there. So it's possible to grow these cells in culture. So what we asked was, are there subpopulations of lamina cribrosa cells, and how do they change in glaucoma? So primary cultures were prepared of lamina cribrosa cells from donors without ocular disease and those with glaucoma. And part of the reason I wanted to just show you this study was because we used genetic demultiplexing using the demuxlet algorithm published by Kang et al. So what this uses is the genetic difference between the individuals in the study. So the cells from the different individuals are pooled together, as you see in the center of the diagram of the right, on the right. And then whenever we do the sequencing, as well as counting the number of genes, we can also look at the SNPs that vary between the different individuals. We also perform bulk RNA-Seq from the same samples so that we knew which SNPs related to which individual. And this allows you to demultiplex the sample on the bottom right-hand side there. It's possible to do something similar to this with any samples now using so-called cell hashing techniques, or the CellPlex protocol from 10x, where you artificially label your different samples. So this approach has several advantages. First of all, it allows you to spot doublets. So that's where we have more than one cell go into the same droplet. And we obviously want to remove those, and that can be quite difficult otherwise. But if there's a mixture of SNPs, we can throw those out. Also, because you're pooling all your samples and running them together, it reduces batch effects. And perhaps most importantly, you can reduce the cost by just running one suspension containing several samples. So the initial clustering here just demonstrates how it's easy to spot the doublets, in this case the green cells. So this is where some cells are stuck together and we're getting more than one in a droplet. So we can remove those and repeat the clustering. And we can see that in this case we got 4 principal cell clusters. And then if you look at how the individuals are spread throughout these clusters, so the different colored dots on the right-hand side indicate cells from different individuals. So we've got a spread right across the different groups, as you might expect. But the cells from the glaucoma patients seem to be focused primarily in one cluster, which seems to have some gene expression suggestive of being a kind of an activated fibroblast-like phenotype. So then just to summarize what I've told you and make some conclusions. So the diabetic retinopathy study using a mouse model of type 1 diabetes, I think is a nice illustration of how this technique allows you to isolate individual cell types from a complex tissue, which is just not possible any other way. Just a note to remind people that the power to detect differential expression obviously depends on the number of cells. So this is something you need to consider in your experimental design and potentially try to enrich for certain cell types. The most obvious findings that we discovered in this study were the metabolic changes with mitochondrial genes and also protein synthesis. But there are many other cell type specific changes that we're now looking into. And just also a note that depending on the system that you're looking at, it's worth considering alternative splicing. I think this will be something we'll see more of in the future, and there definitely seem to be some things going on in the vascular endothelial cells in the retina. So in the AMD study looking at RP cells in culture, this really clarified the system for us and showed that there were subpopulations present in cell culture, which these single cell experiments have now enabled us to interpret the experiments more effectively and also have insights into the in vivo phenotype. So in terms of glaucoma, I think this is interesting that the subpopulations of cells persist in primary culture of the lamina cabrosa, and the preliminary results suggest this may provide some insights into the glaucoma phenotype. So just to give an overall conclusion, probably everyone realizes already that single cell RNA-Seq obviously provides unique insights not otherwise possible. But each study you do is different. So before you embark on a new study, you really need to think about the hypothesis and the question that you're asking to guide the experimental design, obviously. I think this is a very exciting field. We've had so many breakthroughs already just with the technology I've talked about. And I think you'll be hearing from the other speakers about the developments of multi-omics, looking at protein at the same time on spatial transcriptomics. And this is certainly something we'll be doing now to further the studies that I've described to you. So just to finish off with some acknowledgements to the members of my own group and Professor Tim Curtis, who's worked closely on the diabetic retinopathy study, Imre Lengal on the RPE work, and Colin O'Brien from University College Dublin, who has been doing the work on glaucoma. So thanks very much for your attention.